Yeah, thank you for the introduction. We heard a lot about MAR for drinking water production, but in this case we will use MAR for irrigation. That's why it's called our project Agri-MAR, so Agricultural Managed Aquifer Recharge. And I want to point out why it's actually so important, because actually we need 70% of our freshwater resources for irrigation. And that's why, because 40% of our crops are irrigated, and if you don't irrigate, you have a high crop loss. So it's really necessary to irrigate. But on the one hand, you need a lot of water, and on the other hand, you have fresh water scarcity. And we have our study area in the Netherlands, where you have brackish groundwater, so such as in Australia also, and in all coastal areas or delta regions. In a map where salt water was modeled, you can see in milligram per liter you have like 1 gram per liter of chloride concentrations in the shallow aquifers. Also, like last year, we had a very dry summer, and even though it rains a lot in the Netherlands, and you don't think we have a problem with water, but then we had high drought that the farmers didn't have any water available anymore, and it continues, so all the groundwater could not yet be replenished. So like this article in Germany now, that farmers after the dry winter still don't have enough water. And there's also a card for soil moisture, so you can see all this orange areas. There is already now in April 2019 soil moisture deficit, but we are in spring, so it actually should be enough water now available. And another problem, here you can see again the water availability in the Netherlands. So actually there is everywhere water next to the fields, but the problem is this available water that could be used for irrigation has not a good water quality, because it's contaminated with plant pathogens. So the farmers are not allowed to irrigate actually with this water. So this is a map where water samples are taken, and you can see the green and red points. And where there is the red points, this bacterium, Ralstonia solanacearum, which causes brown rot in potatoes, has been found. And then all these areas in the red, it's not allowed for the farmers to irrigate. So they don't have groundwater because it's brackish. They don't have surface water because not allowed. And if it's dry, we don't have water from precipitation, so they just have to face drought. And therefore, and here are also some symptoms of these bacteria. So I selected 3 plant pathogens that are present in the surface water in the Netherlands, but also in other countries. And then you can see if there is an infection, you just have a much lower crop yield. They are causing soft rot, and here blacklead, and this is potato. So you can also see that these bacteria, they don't only infect like potatoes, but also peppers and flower bulbs, which is one of the cash crops of the Netherlands. And we propose managed aquifer recharge as a local scale freshwater supply for irrigation. And we collect the water that comes from precipitation surplus via tile drainage system. So nearly all the fields in the Netherlands are drained like this. So we have drains that lie below 70 centimeters or 50 centimeters below under the field. The water, as excess rainwater, goes from the field down, and then we collect it from there. And normally it's discharged into the canal, but we collect it and infiltrate it into the MAR system. So we displace the brackish groundwater with our fresh water. And as we have a ASTR system where we also have the infiltration well spatially separated from the extraction well, so we have this additional soil passage, which is an extra treatment step, and where we hypothesize that the bacteria are removed. And from last year we have this result from the pilot site where is already a MAR system installed, and the farmer could use irrigation water, whereas on this field there has not been irrigated, and you can clearly see the difference. So for the farmers, this means a lot, because it's just money that is lost here. So it could be a good investment for the this MAR system. Yeah, but we know the water quantity is enough, but in this case we need to find out what happens actually with the water quality. So if we pump down water that may be infected with plant pathogens, this happens during heavy storm events. Like here, you can see that surface water, like here in the canals, mixes with then the drainage water when it infiltrates. So we have here the drains, and we would infiltrate into the Mars system potentially contaminated water. So what happens if this water is contaminated with plant pathogens? We pump it down. Do the bacteria survive this? Like now, how high is the attenuation rate in our Mars system? So such as Declan just showed, when you have a Mars system with wastewater, how fast do the bacteria die here? I want to know what happens with the plant pathogens, and is it safe to use the water for irrigation? Because I, of course, don't want to have a crop disease after irrigating with smart treated water. Also, the farmers may want to use the canal water directly, so this would be an additional option just to treat their surface water and then have always irrigation water available. So my hypothesis is that MAR increases the water quality through the soil passage, and therefore I study the fate of plant pathogens during MAR, and in the end I want to do a risk assessment of the whole system. So what is the risk if there is a contamination, if we have the soil passage and in the end use the water, is it safe to use MAR treated water in irrigation? Therefore, I have an experimental approach where I want to simulate the aquifer conditions in the lab and later on also on field scale. So I start with batch experiments where I just have a bottle with the water and then I inoculate my bacteria. In the Mars system, I have different water types. Therefore, I went to the pilot site and I collected rainwater, but also this anoxic aquifer water, which after the infiltration, of course, is kind of a mix between the drain water, but also the natural aquifer water, or at least this freshwater bubble that we already established. I wanted to know what is actually the influence of the microbiota that is present here and here. Therefore, I treated both waters in 3 different ways. So the anoxic water, I just tested the natural conditions, so no treatment. I filtered it where mostly all microorganisms are removed. And here we have the autoclaved water, which is kind of artificial. There's still a lot of nutrients because I just autoclave it, and everything that is in there could serve also as nutrients for my pathogens. And after these treatments, I added my bacteria, and I tested 2 temperatures. In the aquifer, it's quite stable, 10 degree and 25 degree, because we could also infiltrate higher temperatures and also to have a good temperature comparison. And what happens? How long do these bacteria survive? And you see here the decay from 10 to the 4 to 10 to the 1 colony-forming units per milliliter. This is still a high concentration because in the canals you find maybe 10 to the 4 colony-forming units per liter. So instead of milliliter, but I wanted to, for the detection limit is 10 to the 1, so I had to increase it a little bit. And as you can see, the survival is here up to 100 days. Actually, these experiments are still running, so after now 300 days, they're still happily alive. And just this one bacterium, Ralstonia solanacea, which causes the brown rot, is sensible to this low temperature. And after 40 days, it was not more detectable. And then you can clearly see a trend when you remove here when there is only the filtration. So there are still some microorganisms present, and also the nutrients are not so abundant as here, because here I autoclaved it. And here the bacteria only survives for circa 50 days. And then you can also, the next one, the influence of the microbiota clearly visible in the natural system, which would be like the aquifer. So if the bacteria would enter the drain water, they may only survive for up to 21 days. And here is the anoxic aquifer water. And I was a bit surprised because I thought actually, yeah, it's anoxic, so the bacteria don't have very optimal conditions. But I knew that these two bacteria, they were also facultatively anaerobic. So I knew that probably they would survive, or could survive as long as the other in these experiments. But also this one turned out to be, yeah, quite persistent. But also here, of course, the microbiota here is different than here. And I think here, because it's an anoxic water, they are just more competitive, whereas in anoxic conditions, you quite have some specialized organisms. And I think, yeah, then these were just better competing against these bacteria. And like Declan also just showed, yeah, to get the right inactivation rates, you need also a good model. You cannot, so here you have maybe a linear curve. This is from 10 to the 4, but this is in ln, the natural logarithm scale. And you have a 40 days survival of this bacteria. Whereas when you have the another bacterium, same conditions, and you can clearly see there's first a plateau phase and then it goes faster down. And therefore, we choose to use this Weibull model that is a non-linear model to also account for such a plateau phase. And we also think about extending this because in some of the results, we also have like a tail. And yeah, if you have bacteria that survive at very low concentrations, but for quite a long time, this can also pose a risk if they're still present. So yeah, this is ongoing and then will be used in the risk assessment. And what I'm also doing are column studies. So I'm studying the interaction of the bacteria with soil matrix. So here you have the die-off in the water phase, which I'm testing with the batch experiments. The bacteria could strain, so this is just a physical, they just are blocked between small soil pores, and these are the physical parameters with the water flow that they go through. And you have detachment and attachment to the soil grain, and of course the die-off when attachment. And in the lab it looks like this. So I have inflow water that I continuously purge with nitrogen to have anoxic conditions. It's going, it's pumped through the water. It's saturated. So that's why from down to up to really avoid any pores. And here I have conductivity and oxygen meter. And in the end, I want to know with, and I sample the bacteria here. And for a salt tracer test, I can just sample here. And in the end, I want to get the values of the attachment and detachment rates of the bacteria. So the inactivation rates I will get from the batch study. The flow parameters I am getting from the salt tracer test. And then, these two, the attachment and detachment from the column studies. And actually the attachment and die-off in the water phase are the main attenuation processes in during the transport. And if I know how the bacteria are transported, I can tell something about the fate and include this into the risk assessment. As an outlook, next year I also want to make this in a field scale. So we have now this pilot site which has ISMAR system for 10 hectares of irrigation field. But we also have a monitoring well. So this is now 7 meters distance, and we have a monitoring well here at 1 meter. So I will inject my bacteria, and after 1 meter, try to recover them. And to make a risk assessment, I need to know what is actually the infective dose of the bacteria. So how many bacteria do I need? So in this case, this bacterium to have an infection in here the flower bulbs or in potato plants. Because it could be that only 1 cell per 10 liter causes a disease, or 1 cell per 1,000 liters, such as in the drinking water when you have the E. coli restrictions. All this, I want to use all the data for the quantitative microbial risk assessment, which is a combination of a lot of probabilities, but using the data I generated here. And this is the basis for water safety management to get this ISMAR system in the legislative process, and that this is allowed. yeah, I want to choose as my results I want to use to get the optimal MAR design. The die-off of the bacteria will determine how much time actually the bacteria need to stay in the aquifer, how they are transported. Also will give me an insight of the travel distance. Do we need here 7 meters, or do we need a shorter distance between infiltration and extraction well? I want to use a risk assessment for marine aquaculture, which hasn't been done yet, only for wastewater. And and this research is needed to start changes in the legislation and to enable this SMAR system in the Netherlands for irrigation purposes. Thank you.
So thank you for good presentations. My question is, you just did the experiment in the laboratory, just a column such as. But in fact, if you do the experiment in the field, you find something, because the drain system is very shallow, just 70 centimeters. Yeah. So if the rainfall reaching some fertilizers and pesticide into the drain system, then you recharge this water into the shallow groundwater. Then you put irrigation. So some contaminant pollutants will into the shallow groundwater. So this is a key problem. I think it's.
And this project is together with my colleague also Emil Krausdijk, which is he's addressing exactly these problems. So he is checking how fertilizers and pesticides are degraded during the MAR process. So he's doing the chemical water quality, I'm doing the microbiological water quality, and in the end it will give the whole picture of this local scale MAR system.
Yeah, I hope the field study will prove your method is very good. Thank you.
Yeah, thank you too.
I'm very weak in biology, but wouldn't it be an option to add additional microbes that could compete with the ones that are pathogens? So harmless microbes that could just survive better and that are better equipped against anoxic conditions?
You could do this, but then you have again a legislation problem, because then you have like a bio-augmentation, and this has another restrictions. But I think the biomass in the drain water, but also in the aquifer water, is quite high. So as you saw in this experiments, just in the batch, there's already only like 2 weeks or this 40 days, but this doesn't include yet the column transport. And I think the bacteria will also be removed by the column by the soil passage, and then you have this attachment and then die-off by attachment, and together with the die-off in the water you will reduce again the survival time. So I'm quite positive that actually the survival time will be quite low. And then you can tell the farmer you can infiltrate and leave it for 1 week, and then only after 1 week you're allowed to use the water for irrigation.
Thank you for a very good talk. I have 2 questions. One is, what kind of soil did you use for the column experiment? Was it the aquifer extracted from the field site? And another, if you are planning to add the pathogens into the field site, are you not afraid that it will permanently contaminate the aquifer?
For the first, so I will start with very clean conditions, so that I just use a clean sand, like normal quartz sand, because the aquifer where we are working with is also just quartz sand with a little bit of clay also added, and I will use the same grain sizes. But this is just to have really the transport parameters, really, because you have too many parameters if you measure everything at the beginning. But now during the drilling operations, I also collected soil from the field, and then I also will use this and compare. And I will also use different flow rates, adding humic acids because on organic matter, because they compete with bacteria for attachment sites, for example, then you would have again a reduced attachment. So yeah, but this I just started now, the column, and yeah, it's ongoing.
Okay, so there is no competition with other microbes yet?
At the moment, not. But of course, if you have you have sites that are already blocked with other bacteria, then it could be that these bacteria just pass with the water flow, and then you need again more time, more residence time until you can reuse it.
And this contamination issue of aquifer?
Yeah, we have to convince the farmer, but first of all, this is just the monitoring well will be only like 1 meter away, and yeah. I also will use the column experiments to make already a prediction. And if this doesn't work with the plant pathogens, then I will also use E. coli maybe as an indicator mechanism that doesn't have an effect.
Thank you.
So if you apply your artificial recharge, you get water back which is nearly free of turbidity. What about using UV for disinfection to get rid of a problem with the bacteria?
Yeah. So now our approach is to not treat the water further because it's just additional cost, and this is like local scale, so that the farmer already has high costs to just invest in the ISMAR system. So best would be there's no necessity to use UV, but as I'm in the sanitary engineering department, the people are working a lot with UV and ozonation, so I will get into this also because I received this question now several times already. So yeah, I think it's important to also check this. But it's expensive because you need a lot of water for irrigation.